Okay, primary cell line. Okay, the 2019 past paper, this is starting the multiple choice. Okay, primary cell lines have, right, so this is a definition that we're looking for. So primary cell lines are from normal tissue, and because they've come from normal tissue, it means they will have a limited number of cell divisions. This is the Hayflick number, straight definition stuff you need to know. Okay, question 2. The proteome is larger than the number of genes in the genome of an organism because. Now, you could actually answer this at higher. Okay, so if we have one gene, but we have lots of exons in here and introns, which we're obviously going to cut out, if we do alternative splicing to give us many different mRNAs from just that one gene, that will give us, instead of one gene giving us one thing, we've got one gene giving us 10 or so, whatever. Okay, so it's alternative splicing. It can't be post-translational modifications for multiple mRNAs. Post-translational modification is of the protein. Okay, so we're looking at the number of genes, so that's why it's not that one. Not all genes expressed as proteins would hardly mean that the proteome is going to be larger. And each mRNA translated by many ribosomes, that would give you the same protein over and over again. So it wouldn't give you a larger number of proteins, it would give you a larger number of different proteins, which is what we mean by the proteome. Okay, so C. Question 3. We're looking for a neuron in a squid running from the brain to the tentacles, 0.9 meters long. A 25 millimeter length is depolarized every 0.001 seconds. The time taken for an impulse to travel the length of this neuron. I thought this was actually okay once I'd read it through a couple of times. The first time I was kind of looking at going where exactly they wanted to go. But it is actually as simple as this. 25 millimeters takes you 0.001 seconds. Okay, I have a 0.9 meter, which is 900 millimeters. And then I just did straight proportion. So 900 divided by 25 times by 0.001 gives me 0.036 seconds. That's an S, not a 5. Okay, which is D. And that's it. Okay, I'm going to go at a picture from a synapse just to kind of remind you what's all going on over here. The list shows some events that occur in the cell membrane of a neuron during nerve transmission. We've got binding of the neurotransmitter to the neuron, closure of voltage-gated ion channels, opening of ligand-gated ion channels, opening of voltage-gated ion channels. Which events contribute to the depolarization of the resting potential of a neuron? So basically what stops charge building up inside the neuron? And so what we're looking for here is, right, well, first off, this must cause depolarization of the neuron. When the neurotransmitter binds, that's what triggers the kind of wave of charge being kind of used up. Okay, and then we're looking for things which will, again, prevent a build-up of charge. So anything which opens up channels, it will be that. Okay, and so D. Which row in the table describes the expected effects of inhibition of the sodium-potassium ATPase pump? Okay, this is in a lot of papers. We have our intracellular on this side, and we have our extracellular on that side. Okay, which row in the table would give you inhibition? Okay, so inhibition of this whole thing. So if I block this doing its job, what is going to happen to this? So our sodium ions, if we think about it, are going to build up on the inside because they're not being pumped out. So they are going to increase. Okay, our potassium ions, okay, are not going to be pulled in because they're not being pulled in because this is not functioning. So this is not happening, so I'm not getting potassium moving into the cell. So again, we have a decrease, which actually gives us that's not quite our answer, sorry. And membrane polarity, now that's an odd one to kind of throw in there. But what we're looking at here for polarity, therefore, is a difference in charge. And difference in charge has to be attached to the fact that I have pumped out 3 pluses and taken in 2 pluses in a normal function. But our membrane polarity is not going to be as good because we don't have the change or difference between the two. So it's a decrease, and that's C. That's a reasonably tricky one. Question 6. A decrease in the activity of glucose symport proteins in the cells lining the small intestine could be caused by an increase in. Okay, so if you don't know this, then you're going to struggle to get even off the mark, okay? Glucose is in symport with sodium ions. So that means that both of these are transported at the same time together in the same direction across the membrane. Okay, so what we're looking for is something that could cause a decrease. Okay, so a decrease in the activity of the proteins caused by an increase in something which prevents this working. Okay, so glucose concentration inside the small intestine, if that increases, this would give the glucose an increased concentration gradient. So that's going to help rather than cause a decrease. Sodium ion concentration in the cells, now that's going to impact, because the bigger my sodium ion on this side, the side where I'm trying to get it to, then the smaller my concentration gradient, and the smaller my concentration gradient, the less of a push I'm going to be able to bring to it. So this I like. Okay, C, can we just get rid of? ATP concentration, this is not active transport, so that's not involved in that. Potassium ion concentration in the cells is not to do with the sodium one either, so it is the sodium ion concentration. And that's it. Okay, question 7, it's another number crunching one. Surface area to volume ratio of a cell is an important factor affecting transport. Okay, that's why there's a maximum size of a cell. Surface area to volume ratio is the surface area divided by volume, not surprising. Okay, surface area to volume ratio of an E. coli cell is 4.5. A eukaryotic cell has a surface area of 1,809 micrometers squared and a volume of 7,235 micrometers cubed, compared to E. coli. Right, so first of all, I'm going to have to work out my surface to area volume ratio. That's not particularly difficult, so this time I'm looking for my eukaryote. So 1809 over 7235 gives me 0.25. Okay, and it's asking you how much bigger or smaller is it according to this one. Well, so my E. coli was 4.5. My eukaryote is 0.25. I would just, to get a ratio, just divide them both by 0.25. That's 1 and that's 18. So overall we're saying it's 18 times smaller. Structure question for proteins. Alpha helices in proteins are stabilized by, I'm afraid you just need to know this. Okay, you've got your two types, alpha helixes, which have stability kind of across them by hydrogen bonds, and your other ones are beta pleated sheets, either parallel or anti-parallel, again stabilized all with the hydrogen bonds. Question 9, you'll notice I pulled another thing for this again because it's one that I think is easier to see in a picture. Which row in the table describes the effect of an increase in temperature on hemoglobin and oxygen delivery? So if I just spin this down a little bit, okay, here's the effect of temperature on this one. So as I increase my temperature, so going down these lines here, I'm having a decreased affinity of the oxygen, okay, which means that actually that I'm going to drop off more oxygen rather than hold it in. Okay, so what I'm looking for here is decreased affinity but an increased delivery to tissue. Okay, just checking where we are for that one. Yeah, definitely C. Okay, question 10. A lot of reading in this, but actually when I looked at it again I was like, it's actually not that bad a question. Okay, lipid bilayers of cell membranes contain a number of different phospholipids which are present in different proportions and distributed unevenly across the extracellular and intercellular faces of the bilayer. Figures 1 and 2 show proportions and distribution of 4 phospholipids. So I have here 4 different phospholipids, all with big long names. And then figure 2 is where they are. So SP is pretty much on the extracellular face almost entirely, tiny bit on the intracellular, whereas PS is completely on the intracellular face. Okay, we then have a key. The key can be used to identify each of the phospholipids, and it says which letter represents, I'm not even going to say it, sphingomyelin. It's myelin at the end. Okay, right, so let's go look for that. This is SP here. It's about just under 25% of this, and very much, that was the first one we saw, very much on the extracellular face. So first question, is it more than 25 or less than 25? I've just said that it is less than 25, so I'm going to 3. Is it found mainly on the extracellular or the intracellular? Very much on the extracellular. Answer is C. I see it's a lot of reading, but in reality that's not a hideous question. Short-term variations in the time interval between heartbeats is termed heart rate variability, HRV, is one measure that can be used to assess health. Box plots give information from one study about the influence of age and gender. So we've got males and females, and then you've got a box plot showing the median, the lower quartile, the upper quartile, and the maximum and minimum values. Okay, from the data shown, which of the statements about HRV is correct? I must admit, I didn't particularly like reading through this one, but the nice thing is that it's the first one, that you get to trend towards lower HRV in in females, and so this is our female line, and at each one you would say that it's actually a decreasing trend. Okay. The other ones are not true, but some of them are pretty close to where you would expect them to kind of be the line, so just be very careful with that one. Okay, so same data that you had for question 11. You're asked which row in the table describes features of the study. So the type of data that was collected, was it ranked or continuous data? Since we're doing a number, that has to involve continuous, okay? And because we have got several different things in here, we're looking at males, females, and we're also looking at age, it is multifactorial, so D. Okay, question 13. There are approximately 40 species of birds of paradise in New Guinea, on islands nearby and in areas of mainland Australia. They are thought to have evolved from a crow-like common ancestor that lived 20 million years ago. The list describes processes that are likely to have contributed to the evolution of different species. So we have X, food availability on a certain part of one island favoured the survival of male and female individuals with slender curved bills. Before I even get to the question, you should be recognising that's natural selection. On one island with abundant food choices, females choose mates whose head feathers have elongated plumes. And again, before we even get to the question, you should be going, well, that's a form of sexual selection because of female choice. And then Z, some males and females of a species of crow-like mainland bird were blown by a freak storm to some of the islands. So that's something that's not a form of selection. That has been a random, not selected part of this. So because you were just wrong place, wrong time. So that is genetic drift. So which row matches up with each one? So we've just said basically Z. Z is genetic drift. Okay, so here Y is sexual selection and X is natural selection. So correct answer is B. 14. Which row in the table indicates factors that can all lead to a high rate of evolution? Okay, so for a start, selection pressure, we need it to be high. Okay, the higher the selection pressure, the quicker we're expecting there to be a push for evolution. Our generation time should be short. Well, they've pretty much decided where we're going with this one now. Sexual reproduction will obviously increase rate of evolution. Horizontal would have been reasonable to put in as well, but they were asking for which one indicates that it can all lead to, so it has to be D. Okay, question 15. Which of the following adaptations can be explained using the Red Queen hypothesis? So this is co-evolution. Okay, I need to have two things which are pushing each other in their own evolutionary setup. Okay, so acceleration of cheetahs related to the speed of the antelope they prey on. This is a predator-prey relationship. I'm liking that one for Red Queen. Okay, migration of insectivorous birds is influenced by the availability of insects. That's just a food supply thing. This is not the insects changing because of the birds. So no. Resistance of insect larvae to parasitic wasp infection is a response to these wasps. So what we've got is resistance of the larvae, and then we've got the infection of the wasps. So we've got both of them are in an arms race to try and see who's going to get there. So that one is Red Queen. Ornamental head plumes of some male birds are attracted to females. That's a little bit of a random one. This is entirely about sexual selection. Okay, so our answers 1 and 3, and hence B. And that's you.